anything. So we're just like live. Pretty cool.
We were not live, and now we are.
Now we are alive. Ooh, we're back alive. Dead alive. So I got that book that Dr. Chris Thompson recommended,
Oh, okay.
Complex Adaptations in Evolving Populations. And of course, I read six. Oh, shit. Polish. You couldn't hear that, right?
Oh, what?
You didn't hear anything. Basically, Google Chrome is not working. Their audio quit working with my Mac.
Oh,
I don't know why, but now Firefox does. But anyway, so
hmm.
yeah, Complex Adaptations in Evolving Populations by T.H. Frazetta. And it looks like he's got he I guess he talks a little bit about this thing you were talking about with basically genes that have more than one effect, right? They attach more than or they have more than one phenotypic effect. So when those genes mutate, they could fuck up more than one thing. Like if they mutated to your advantage, pleiotropy. If they mutated to your advantage for one trait, it may mutate to a disadvantage for another trait. And it turns out there's a paper. I don't know if I found it or if Dr. Thompson found it. That let me see if I can find that by Augie, Pleiotropy in Developmental Regulation by Flowering Pathway Genes. Is
Hmm.
it an evolutionary constraint? And let's see if I can put that in. I better at least put that in the description. Let's see. Oh, let me.
Hey, Dave.
Hey, Dave is here? Oh,
Yeah, Dave's in the side chat.
that's okay. Two Daves now.
Huh?
There's
There's two Daves?
David Lori David. You know, the one that always comes on is Lori. Uh, yeah. Anyway,
Well, Dave and David aren't I don't know. I don't consider them the same.
Snicker bars for Bigfoot Mike. He will clear up that internet thing. All hail Bigfoot. Okay. Anyway, so this paper. Did I say that? I did. Let's see if I can. Let's see if I can. We can actually see that paper. That would be nice. Ah, there it is. Okay. So this paper. Whoops, that doesn't work. I don't like that
Oh,
shit. Oh, how about that shit? Okay.
there we go.
Yeah, this one. So basically, consequently, it is considered a constraint on the evolution of adaptive phenotype because of potential antagonistic selection on correlated traits, or alternatively, preservation of functional trait combinations. So it goes through and it talks about that. And one of the ways around that is for there to be a duplicate, yeah, a duplication of a gene, and then one gene basically keeps performing the function that it did before, and the other
Right.
gene, and then you're down to okay, if it changes the regulation on the gene that was duplicated and creates a beneficial effect, now it's decoupled from the original trait, so it wouldn't have an
Right.
effect. So that's one way out. And they're talking about molecular biologists have have embraced the phenomena of pleiotropy less enthusiastically. And the reason for that is because it's very difficult for them to uh, research those kinds of genes, the genes that have more than one effect.
Right.
But in developmental biology, there's a whole like there are genes like the Wnt pathway that you'll find in the Drosophila or the fruit fly, and you'll find them in human beings. And the Wnt pathway has been preserved basically across all animal life. And those are genes for which that have effects in embryology or morphogenesis, and
Yeah,
they
well,
have tons of effects on
long-standing
everything.
effects. Yeah.
Yeah. So those things are fixed, and those aren't how changes are typically going to happen. So anyway.
Oh, that's what the paper says?
Well, the paper, I haven't read that much of the paper, to tell you the
Oh,
truth.
okay.
Yeah. But let's see. Well, we can go down and let's see if they got a conclusion.
Well, what do they say based on what you've read so far?
They said one of the ways around it is gene duplication. So
Right.
that was the first thing I ran into. And let's see. Okay, we have conclusions. Okay. The flowering time pathway in Arabidopsis thaliana is one of the best characterized genetic pathways in plants, yet new pleiotropic functions of its genes are being discovered all the time, even though patterns of population genetic variation reveal evidence for natural selection on these genes. So I think they're concluding that it doesn't have as big effect as you would think. So there are variations. What they're saying is there are variations in those genes, even though they have pleiotropic effects. They affect more than one thing.
Okay.
Which is interesting. Even though patterns of population genetic okay, let's see. What did I read that? Reveal evidence for natural selection of these genes, interpretations of their functional and adaptive significance will need revision to accommodate the different pathways of direct and indirect selection acting through multiple traits that a single gene may regulate. Organisms developing in the wild, moreover, may express even more pleiotropy than can be detected under controlled conditions in a laboratory, as indirect pathways of pleiotropy operate through environmental interactions, whereby one trait may alter the environment experienced by subsequent life stages, which in turn alters the expression of plastic trait expressed in later stages. Okay. What do they conclude? Do they conclude may contribute to the divergence of gene functions? I don't know. Anyway, the paper requires a bit of work to read.
Okay.
It's a long one. It's about 12 pages.
Oh, okay.
Yeah. And we know a lot about Arabidopsis. What the hell is it called?
Arabidopsis.
Yeah. Why did that sound weird to me? Arabidopsis.
Yeah, whatever it's, however it's pronounced.
Every now and then you say a word and it's like, God, that word doesn't sound like a word. I have that problem occasionally.
Yeah.
Anyway.
Wait, there's a new Aaron Raw video.
What's that now?
I said, Top Dog says, did you see the new Aaron Raw video?
No, we didn't. You should link that. So, you had a little exchange with a climate denier last night. So how did that go?
It went about as expected. Gur eventually gave me a wrench, and I posted some links, and for some odd reason he didn't, or the skeptic didn't post any more comments after that. I wonder why.
Well, maybe he was off reading those links and changing his mind. Could that be?
Well, he must have been doing it for an awfully long time, because he never popped back in.
Yeah. Let's see. One of your links is climate change, global temperature. Yeah, I'm looking.
Oh yeah, and so I showed one of the graphs when I did go in the room, and even Gur was surprised by some of the trends in the graph, which made me happy.
Yeah.
Or not happy, but like gave me pleasure in teaching him something new. You know what I mean?
Yeah. Yeah. So he says, chosen one in the kit. Andrew, plan on blocking me? That's what he asked.
Oh yeah, he thought since I was asking for a wrench that I was going to block him.
Oh yeah, okay. Wow. Chosen one in the kit. You are shooting blanks. What else does he say? Andrew, so far you cannot provide a link. And then you provided him a link and then he went silent?
Yeah, so how it went down.
Yep. Okay.
Okay,
Interesting.
apparently Captive Desk is also met into a global cooling guy. Oh, is that someone who thinks, I don't know, can you elaborate, Captive Desk, please?
I ran into a scientist said global cooling guy, citations aplenty that I posted, not
Oh,
another
okay.
peep.
Someone who said the scientists were predicting global cooling. Okay, I
Yeah,
see. I misread that.
not another peep. Yep,
Well, do we have his email? Maybe he could pop in and
I do.
discuss that.
I do. Except, God damn it. Restream doesn't seem to have a send email invite, but I can figure this out, can't I? Can't
I
I?
hope so. You might have to do that a few times coming up
Invite
if you're gonna
guests.
go with
I know.
Restream.
I know. I do. Okay. How can I invite guests? Okay, copy. I'll see what I can do. I will work
Yeah.
on that.
Yeah, if you figured that out, please come and join us, Captive Desk.
Yeah,
Always
that'd
a
be
pleasure
great.
to talk to you.
Oh, oh no. I gotta go look up the goddamn email address. What the fucking fuck?
Well, while you're doing that, I guess to describe things a bit more, I feel like maybe one thing I could have done a little better was not being so uh, condescending, I guess, because like he was, I understood where he was coming from, of course, but I maybe could have been a bit less mean about it.
Yeah,
He may have responded a bit better if I had done that. I don't know.
I don't have captive death's email. I don't know
Oh,
why. At least I can't find a clue by just typing it in, and I didn't write it down. So I'm gonna need an email, man. I could put the link out there. You know,
yeah,
people
might
can
if you're okay
people
with that,
people can join us.
might as
I don't
well.
know how many. I have no idea how many people can come in on this thing. Uh,
Yeah, that's a good question. Uh,
I think that's it.
Probably the same as StreamYard would be my guess.
I think that's the link.
So yeah,
Anyway,
anyone who wants to pop in, please come in.
hey Randy, KCA Randy,
Hey, Randy.
and yep, 5 whole watchers. Oh my God, we're real. Oh, the Uncle Jesus show. Professor DK and Associate David Keith, Solar Engineering, Harvard University, and the Sky Business
Oh,
for
did we
Beginners.
already attract someone?
Professor DK and Associates. Oh, the
Uncle
Uncle
G.
Jesus show. I think that's that really cool. I don't know if that's a channel I'm thinking about.
I
Not
haven't
sure.
seen him around before, so he must be new.
Yeah. Let's see. Hide message. Oh, this works kind of like StreamYard. This is pretty cool. Anyway,
Are
yeah,
you saying it's StreamYard but better?
but better. Yeah. For one thing, they have a black background, so it doesn't highlight my face and make me look like a ghost when my camera's on, which is kind of nice. So I don't know. Anyway, so I've been digging into these little guys called echinoderms,
Ooh,
and
okay.
yeah, and they are pretty awesome looking little critters. Starfish.
Echinoderms, weird and wacky and so interesting.
Yeah. So what does that cover? Echinoderms. Which kinds of animals that people are familiar with? Sea urchins.
Yeah, sea urchins, sea stars, sea cucumbers,
Yep.
those things that look like plants but aren't actually plants, sea lilies. Um,
Pokey things.
what else? Yeah, yeah, lots of pokey things. What else? What else? Lots of extinct echinoderms that I can't think of at the moment. Uh,
Yeah.
yeah, those are the major groups that people tend to think about.
Wikipedia is not for sale. What?
What?
Oh, you get a little message up here from. I'm not showing it. You get a little
Oh,
mess.
okay.
You get a little message from Wikipedia.
I was like,
I
why are you trying to sell Wikipedia?
I don't know. The thing is, I already donated, and I don't know why they keep because I donate monthly.
Ah,
Uh, and everybody should. Okay, so these are. This is not showing the whole page. That is a problem. We want to move that up there, and we want to move this down there.
There we go.
And, yeah, now you can see the whole page. Okay. So we got asteroidy.
That'd be the sea stars.
Ophiuroidy.
And things that look like sea stars but aren't sea stars called bristle stars.
Oh, and the what are the blastoids and the crinoids?
Yep.
Those
Yeah,
are those
extinct things.
Yeah, they're both extinct, right? Yeah.
Yeah, or most of them, I think. I don't know if any are still alive.
Yeah.
They used to be like really common, so instead of coral reefs, you would have crinoid reefs.
Yep.
In the geologic past. Hold on.
Is that your doggy? Your doggy's he's gonna bite you. Doggy gonna bite him. Oh, somebody gave me an email. No wonder I couldn't find you, god damn it. You should have your name in your email. That's called giving us a clue who you are. That would be helpful. Let's see here. But anyway, I put the link out there in the. I put the link out there in chat, so you should be able to click on that. But I'll send you an email too. Hang on a second here. Yeah, I'm going to send it from here. Or I think it's the link.
Okay, sorry about that.
Nope. I'll see.
There's a package delivery guy nearby, and of course the dogs like to bark at him.
Yeah, he kind of went nuts, right?
Yeah.
Didn't like that. He growled. I mean, it was awesome.
They're quite vocal.
Yep. Kind of like Snack. Oh, so it's that kind of a day where I don't really know what I'm doing, and I really don't care.
That should be a song.
Yeah, I don't care what I'm doing. I don't
Hey,
care.
Top Dog.
Anyway, let's see. If you can see that, the echinoderms, basically.
Yep.
I can make that bigger for the time being, except, oh my goodness, I have to reorder. Yeah, so you kind of see where they are. There's protostomes, and then there's deuterostomes, and echinoderms are, closer related to us than some of those protostomy things.
Yep. And
then we have the hemichordates, arrowworms, cephalochordates. Hemichordates just have a notochord, right? And cephalochordates, what is different about them, cephalochordates?
Well, cephalochordates are true chordates. I don't know if hemichordates have an actual notochord.
Okay, I think they do, hemichordates.
Well, while you're checking that, I'll be right back.
Okay. Hey, Top Dog.
Hey.
Oh, let's go look at this. Hemichordate. Okay, phylum which consists of tripoblastic, meaning they have 3 layers, enterocoelomate, meaning they have a gut, and bilaterally symmetrical marine deuterostome animals. Boy, is that a mouthful. Generally considered the cystic group of echinoderms. Let's see, acorn worms. So they're acorn worms. Live in burrows. I'm trying to find out if they have notochord. Anatomy, body plan. Okay, dorsal vessel, stomochord. Ooh, what's a stomochord? Body plan. Okay, body of acorns. Worm shape divided into anterior proboscis. Don't have to read that. Contains a pharynx. Perforated with a gill, so they have gills.
So we're looking at sea urchins?
Yeah. Looking at where sea urchins are. And, And now I'm looking at the hemichordates. to find out. Let's see. Okay, the stomal cord. A hollow neural tube exists among some species. So basically, I think that's why they call them hemichordates. Probably a primitive trait they share with the common ancestor of chordata. So they have like what was that again? Hollow neural tube. Not sure if they have a notochord. I don't know where Andrew went. Anyway, so you kind of see where they are on the family tree. And so echinoderms are up there pretty well separated from us. So. But echinoderms are interesting because we've learned a lot about their gene regulatory networks and how they develop. And this guy is, I believe this is the larval stage of the echinoderm. Or no, that's actually a fully developed echinoderm. And they have a pretty simple body plan. They have this skeleton, which consists of these pokey things and usually five magnesium and calcium rich bones, sort of, that spread out like the sea stars will have 5 stars, 5 legs, that sort of thing. And this is like a sea star, except it's bundled up at the top. So it looks like a globe, but it still has, you can still see the 5, basically, these 5 blue things here. And that makes the echinoderm. And Andrew's tending his dog yet. But they have their deuterostomes like us. That means the butt forms first, and then the mouth forms later. And they have a simple stomach. They don't have too much for organs. They have a real extremely small neural net. I think somebody told me, hang on a second here. When we're looking for a simple animal to study embryology or evolution, they're a really good model for us. So, let's see, how much nervous system. They have 5 radial nerve cords running along those 5 legs. Let's see, trying to find some information on how many neurons they have. 2 facts. Okay, true brain, no brain. And I think they have kind of a primitive brain too. So they have a neural net that makes a brain.
Cool.
Yeah. Do echinoderms feel pain? That's one of the Google entries. Interesting question. Yes, they do. Do echinoderms
probably have to have some kind of sensory of danger?
Yeah, do they know that they feel pain? No. To know things requires more neurons.
Not going to same as humans, of course.
Yeah.
How we receive.
Let's see. Elephant, rhesus monkey.
Did you figure out if hemichordates have a notochord?
They have a neural tube that
Okay.
they think is a precursor of the notochord.
Yeah.
So,
Okay.
Yep.
I was saying, like, they probably have a precursor structure, but they're not technically chordates.
Yeah. Yep. Tardigrade. Okay, so starfish. Around 500 neurons for a starfish, but.
Oh, wow.
Jellyfish, 5,600. The sea slug, 18,000. They have a lot. Fruit fly, 100,000 neurons. That's pretty good. Anyway, so what I was looking at is where they're placed in the tree. So they're They're out there. And the thing is, they already have a developing stomach. They have that pattern of a deuterostome.
Right.
So you can figure out all that. And then the interesting thing that you got to learn about embryology is that cells are sticky, and they're little. Okay, they're little. Imagine a little cube of Jello, with a super thin film around them, which can easily be distorted. And the only reason cells stick together is because of the cytoskeleton. And so they have actin mostly on the inside that creates a cytoskeleton. And that gives the cell its shape. Basically, how the actin is arranged in the cell will give it a shape. But cells stick together, and now they stick together with primarily very, very complicated, and there are hundreds of molecules that make cells stick together. But one really important one is cadherins, and they're very important in developing. And this one here has N-cadherin on the left. Green cells have 2.4 times more N-cadherin than red cells. So they have the same sticky substance, but what happens when you mix them up in a plate, like on the left, you mix all those cells up, they automatically organize so that the cells with the most cadherin, the most stickiness, end up at the center, and the other ones end up in the outside. Okay.
Right.
So you can see a real simple trick for embryogenesis is just altering the amount of sticky stuff they have, and then they will stick together. And as it turns out, let's see, we go a little further here, signaling proteins. These are the cadherins. Cadherins like to stick most of the time to other cadherins of the same type. N-cadherins, I think, are in the neurons, and E-cadherins are really important in embryology and early development. And they tend to stick together, and they join. If you can see, I'm not sure how well people could see that, but on the right-hand side, cadherin binding, they'll show that the ends stick together, and it's calcium dependent. That's what they're named for. So they're calcium dependent adhesion molecules, and they have to have calcium in order to stick. But if they have calcium, they will stick to each other. And so these things tend to form like a layer of skin or layer of cells, all of them hooked up and sticking together. And then at some point in embryogenesis, a lot of these cells that are in a layer, okay, like in a tripoblast, which means there are 3 layers, the ectoderm, mesoderm, and endoderm.
Right.
These things, basically, some of those cells will break up, and they do something called a endothelial to mesenchymal transformation, or EMT. And that means that these cadherins suddenly start losing their stickiness, or basically they could lose calcium, or there could be less cadherins, and these cells start to break up, and they'll migrate out from the epidermal layer, and they'll migrate into the interior of the blastula, or the forming organism, and they often become muscles and skeleton. In the case of echinoderms, they become those little 5 little skeleton sticks that start to develop. And I do have movies on this, but
it's interesting that they're not, of course, well, moving under any intelligence or something like that. They're moving because they're chemically signaling to each other, and those signals, they're kind of sniffing out the signals like a dog to where its source is.
Just thermodynamics. At the level of embryogenesis, each cell behaves like its own organism, and it has its own environment and its own little group. And some of them will form a pack of cells, and they'll stick together. They'll group together, and others will be completely independent, and they seek their own path based on their environment.
Right.
And there's So, and then the other thing is there's different kinds of signaling that go out to cells. So you'll learn about endocrine signaling, which would be the entire organism basically gets a signal from somewhere else in the organism. But paracrine signaling is cells in the immediate neighborhood will get a signal from you. So one cell sends a signal to its immediate neighborhood, and you have paracrine signaling. And then you have juxtacrine signaling, which means it'll only signal the cell that it is sticking to, that it's right next door to. And then, of course, we have autocrine signaling, which where the cell sends out a signal and receives it itself. So it kind of creates its own environment. And then there's endocrine signaling, where the signal never leaves the cell. It's just something that happens inside. So there are those kinds of signaling that you'll learn about. Paracrine, juxtacrine, and autocrine are the important ones. And we've got a little picture here of two cells sticking together. And of course, around the cells, you have this extracellular matrix, which the cells themselves create. So they create this basal lamina, this matrix around them, and they exist inside of that environment. And let's see, so there's all kinds of cell signaling. So cadherins are calcium-dependent adhesion molecules on the bottom. And then let's see, pleiotropy.
Oh, that's what it stands for. Okay.
Yeah. Cadherin, yeah. Calcium-dependent adhesion molecules.
I was just looking at the letters, and that makes sense now.
Yep. And they are in prokaryotes as well. They found all sorts of basal cadherins in prokaryotes. So there is an evolutionary ancestor for them in cells that do not stick together typically.
Right. They probably did something else and were co-opted to do what they do now in multicellular stuff. Yeah. Which evolution.
Yep. So here's a picture of basically that's on the upper part. It shows like an epithelial layer or cells that are sticking together, and the cadherins poke through the cell membrane, and then they hook up. So you can see between these two different layers, you see the cadherins on the blue part on the left diagram. Okay.
The green spiky parts.
Yeah, the green spiky parts, they stick together. Okay. And then they hook through vinculins and talin. They hook up to the actin cytoskeleton inside the cell. And the cytoskeleton inside the cell can be basically there's like a layer of skeleton right around the edges of the cell. Sometimes those are different molecules like laminins, and in the nucleus, it's lamins. And then, but anyway, they hook up to these kind of stiff little, if you imagine cotton that you poured nail polish on and it's stiffened. So it's kind of stiff. And this actin is constantly remodeled. And the way your brain forms is really interesting. I don't, hopefully I have a picture on how brains form, but I'm not sure if I do.
Brains.
But, so if you imagine a layer of cells, okay, this, like you see this layer at the very top of the illustration on the left, we're showing the actin, okay? So if the actin in the bottom of the cell starts to constrict along that layer, it becomes narrow, and the top part becomes, how the hell is that? No, the top part would become narrow. I've got to get my orientation right for the embryo later on. Top part becomes narrow and the bottom part becomes fat because of the way actin remodels the cytoskeleton. What'll happen is you'll have a tube form. It'll start to form this invagination. So it'll basically create a little hollow tube. And then in your brain, that little hollow tube joins at the top and you end up with a neural tube. And that is the beginning of your brain developing. So the actin is very important in the shape of the cells, and physics is very important in how cells stick together or don't.
Right.
and what they stick to. So as you go through in, let's see here, I'm trying to, those are the genes, and this is okay, gene B. I'm trying to find a network. I think
Yeah,
I
'cause
have
they can
one down here.
get pretty complex.
Yeah, and I don't have a whole lot of stuff done for us yet on our echinoderm journey, but I have a
Okay.
few things. Okay. So what happens is the cell, is fertilized, and then it starts to divide, and it divides first of all in the upper left, A, meridionally. So it divides basically along the meridian for the
Okay.
first couple of divisions, right? And then it starts to divide equatorially from the 4-cell stage to the 8-cell stage. And now we're on the second row down there. So we got 8 cells, and then 16 cells, and then 32. And then they start, you end up with an animal half of the organism, and you end up with a vegetal half. And they call it that because I think in chick embryos, the bottom part ends up being the yolk, and they call it the vegetable, and
okay.
the top part becomes the animal. But in echinoderms, it's a little bit different. So then you end up, it divides up, and on top you have mesomeres, and on the bottom you have micromeres and macromeres. The micromeres are on the very bottom, and they form a veg 1 layer and a veg 2 layer. And then we have, let's see, what is, I forgot what AN is. AN derived. I think is that, I couldn't mean anus, could it? I can't find it. Anyway, so.
It might. I don't know.
But the thing is, once these cells start to divide, like when they first come out of the mama, there is a gradient from the top of that organism all the way to the bottom of a couple of different, basically proteins that are more heavily available on the bottom of the organism than they are at the top. And that's why it divides differently, and it starts to differentiate. And then it goes down, and it just keeps differentiating and differentiating. And I
Kind of
don't have.
the direction they divide in, so to speak, is also important.
Yes, yeah, and that has to do with the basically the actin or the microtubules on the inside remodeling and dividing in a certain way around the centriole, and there's a whole lot we don't know about in that mechanism, but that determines whether it's going to be an equatorial, or it's going to divide along the equator or the meridian.
And that would presumably affect it in the earliest stages, because like if you change that the direction cells are dividing in, like the heart, for example, that's probably not going to affect it as much as if you change how the cells divide during the blastula stage.
Yeah, that's very important. That's where the differentiation starts. And this is for C. elegans, the worm right here on the left. But it shows you from the zygote, you end up with this unequal cleavage, and then you end up with a P1 kind of cell, and then an AB, and the AB go to the ABA and the ABP, and you end up with a hypodermis and the neurons and muscles are part of it. And then on the right-hand branch, the P1 divides into EMS, is that endothelial to mesenchymal? I'm not sure what EMS is. I'd have to look at the paper. It goes down, and so what you can see is that there's an evolutionary tree of cells. They have an ancestor, and they keep dividing, and becoming more and more basically differentiated until they become the organisms. And this is another diagram.
With the caveat that the genes are actually changing just the expression patterns for the most part.
Right. Yep. Let's see. Let me see what this diagram is. Anyway, we probably shouldn't go any further until I do more presentation. But the important takeaway today is think about cell adhesion. And it turns out that there are different cat herons. I think there are about 100 of them, and we found 180, I think, across the basically the vertebrates. There's like 180 different cat herons, and they tend to stick to each other more than they stick to any other. So they match up, and with that stickiness, so what we have is like if you could buy glue. Where you had an A-type glue, a B-type glue, a C-type glue.
Right.
The A-type glue would just pick up like the dust. The B-type glue would pick up the cat hair.
Or
And
you could have like Velcros of different, I don't know, like what you call it, that just stick to each other differently
yeah,
because they have different,
yep,
the fibers and whatnot.
yeah,
Because I mean, that's what in a loose sense, these extracellular proteins are. They're basically biological Velcro holding all the cells together, and they
right.
can, like you said, be unstuck from each other if need be.
Let's see. Unmod me. Carol the Atheist wants to be unmodded? What do you mean unmod you?
I don't know.
What
She
if,
just doesn't want to be a mod, I guess.
but if you're not a mod, then people can mess with you. I can
Yeah,
unmod you
that's
if you
true.
want. Well, she asked. I'm going to obey. I don't want to unmod you, but I will. It's with great pain that I unmod you.
Yes.
Great pain. So,
Hey, Maya.
oh my. Hi Maya. And let's see, who else we got? Carol the
It's
Atheist, Captain Uncle Jesus Show, Stephanie. Hi Stephanie, our resident poet. It's very important. Uh, anyway, let's see how the brain develops, grows, and learns throughout our lives. That's a good one. Can I open that in a new tab?
it's interesting how it's not embryologically speaking just genetics that determines the
Right.
form. It's also
Yeah.
partly self-organization in a sense.
There's
Like
a
you talked
lot of
about
physics.
the physics behind it.
Yeah, there's a lot of physics involved in it, and that's the important part. And the physics involves the stickiness of the cell membrane, the basically, and that cytoskeleton, what that actin is forming in the cell. So
Right.
actin can make cells like they can send out pseudopods. That's all actin. So the actin like rebuilds at the tip of the pseudopod, the little finger that
Hmm.
sticks out, and it disassembles at the base. And as
Do
it
you know if that's
disassembles,
the same mechanism as in like an amoeba, for example?
yes, it is, and
Oh,
that's the
okay.
cool
I
part.
didn't know that.
Right? Yeah, that's the cool part. That the basically our cells crawl around, especially at the embryo stage. For development, cells do crawl around, and they have pseudopods, and they even
Right.
have some of them even have cilia and flagella, and they move around.
Yeah, they kind of walk around, kind of.
Yeah, but the physics of cells is very important, and then beyond the physics, you have cell signaling. So every cell will have receptors, and the signals that are floating around in the extracellular part, the ECM or extracellular matrix, those signals, depending on how concentrated they are, will tell the cell to create certain proteins, and now it will become a different kind of cell.
Right, and that's kind of
And
where
then,
the genetic component also plays a role. Like if you express more of those sticky proteins, then you're going to get a different response than if you express less of them.
yep. And the ones that have the most expressed will end up at the center of an organ, and the ones that have the least expressed will end up at the peripheral. And you can see how organs can develop. So,
Right.
which is kind of
Speaking of
interesting.
cilia, actually, I recommend. Well, first of all, everyone go check out the channel Journey to the Microcosmos,
Oh
and
yes,
in particular to look up they have an episode on a walking ciliate. So it has cilia, but it doesn't just float around and wave around kind of thing. It actually uses its cilia to walk, so to speak, on So I recommend everyone go check out that and their other videos. Just while I'm thinking about it, they're
See
pretty
if I can
good.
find. So the physics basically, and the membrane receptors and the signaling that's going on from cell to cell. So every cell has its own environment. It'll be inside of this little area that's getting certain signals, and some of the signals come from the maternal zygote, or the egg, and how the egg develops, and that
Right.
forms that forms your head or your butt. Basically, it gives the direction of the cell, and it starts to differentiate in 3 dimensions, and you end up with posterior, anterior, and ventral and dorsal. And then the embryo develops from there. Now, no creationist would deny that everything from the zygote forward happens entirely with chemistry and natural processes.
I don't know. Maybe some would.
Well, yeah, there might be
There
one
probably
that
are some. Never know.
thinks that God is in there. Well, they think God sticks a soul in the zygote,
Yeah.
right? When it gets
Yeah.
fertilized. Yeah. So when it gets fertilized, so like when man and the woman are having sex, God comes into the room and adds his little bit, right? What a pervert. That's just
Oh,
plain
you
creepy.
might
That's
get
creepy.
demonetized for that speed.
That's creepy. That's creepy.
Yeah.
Oh, I'm demonetized as hell.
Yeah. Or some people will definitely take issue with that for some reason.
God's creepy.
Yeah, exactly. Or what Stephanie does.
I think Sal Cordova might be upset by that statement, right? That's one of the things that he seizes on. Like once I said "fuck Jesus" and he got all offended. At least he says I did. I'm not sure if I did say "fuck Jesus." Have you ever heard me say "fuck Jesus"? I don't think I've ever said "fuck Jesus."
I don't know. You probably said at some point.
I wonder if I have said it. Like in this stream, have I said "fuck Jesus" at all? Because I wouldn't want to say "fuck Jesus." I mean, that isn't something I would do ordinarily,
Well, I agree with you, Stephanie. At least intellectually speaking.
God does nothing; you do it. There you go.
No, the thing she said after that.
Oh,
there we go.
Sells a child. Yep. I haven't said "fuck Jesus" even once. So basically, Maya has my back on this. Anyway.
So another thing I want to talk about actually was I believe it hasn't been peer reviewed yet, so you should always be skeptical of those kinds of papers because they need to be scrutinized still. But I guess you should be skeptical about any paper in science. But recently, they actually took some of the most distant galaxies that were previously found by the James Webb Space Telescope, and I think like 4 of them or something like that. So they were between redshifts of 10 and 13. So that would mean that some of them are literally like 300 or so million years after the Big Bang. So really close to that threshold of when galaxies first formed. And now they've managed to get actual spectroscopic redshifts of those galaxies. So for those of you who don't know, there are 2 kind of types of redshifts. There's what's called photometric redshift, where basically you're estimating how far away it is, how redshifted it is based on its brightness, the intensity of the light you're getting, versus spectroscopic redshift, which is your spectrum. You think about like prism and taking sunlight and getting all those colors. It's basically the same thing but more complex. And after getting those measurements, they confirmed that these distant galaxies are it seems that far away. So they're really early galaxies, and they actually measured some of their properties based on this new data. Um, for one thing, as far as I can tell, and again, these are still preliminary results, so more research is needed as always. But the galaxies they did look at were relatively poor in what are called metals. So a metal in astronomy is any element heavier than hydrogen and helium.
Right.
I don't know why they call it that, but that's the name they chose to go with all the other elements. So these early galaxies were relatively metal poor compared to uh, the nearby ones around us today. So the modern galaxies we have around us today, um, and also some of them which appear to be much more massive um, than we were expecting, based on the light they got. And I don't know the exact details behind it, but they actually found that the uh, the star formation rate, of course, was very high. So that's kind of to be expected. There's a lot of gas and dust floating around for new stars to form back then. Um, but they also found that the stars had masses that made the overall uh, galaxy mass distribution uh, consistent, they said, with lambda CDM, or dark energy, cold dark matter cosmology. So, contrary to what uh, some people have been claiming, Big Bang skeptics basically, it initially at least seems that these results are not in conflict with our current cosmological model. So,
Right. So
again,
the
like I
only
said, we always need to do more research, but
yeah, the
so
only
far it
the, o-
seems the right track.
so the only surprise is that the galaxies are there basically formed a little faster than they thought,
Yeah,
but
that
they're
we were able to find them so easily. Like they
right.
thought that we need more time and energy to find these galaxies, but no, they seem to be slightly more common and early on than we thought. So that's
Yeah,
always
and they
interesting.
and they don't look like mature galaxies because they have very little heavy metals and heavier
Yeah,
elements. Right. Yeah.
yeah, and that, and
So
like I was saying, they're also forming stars like really rapidly. So comparable to uh, similar sized galaxy like the large Magellanic Cloud. Um, I think they said it was a bit higher than that in terms of star formation rate.
yeah.
So they
So
are
that was
different than today's galaxies.
the thing that was making them lose sleep and panic is that
Right.
galaxies basically formed a little faster and a little earlier than they thought. So that means that uh, probably says something about the density of matter right after the Big Bang. That it basically
Right.
there was more density than they expected. So their
Right.
So
models
see,
have to be.
kind of
Go ahead.
supports my idea of that black holes were like at the early beginning of the Big Bang, like
Yeah, how would
supermassive
that?
black holes and stuff.
Yeah, that could be possible. So, what do you
I
think
mean,
of that,
we
Andrew?
knew they were, had already formed somehow, like really close to uh, the time of the early galaxies. So
Yeah, and that
we
would
just
drive
don't quite know how they formed yet so
yeah, that
quickly.
would drive galaxy formation, basically.
Yeah.
Yeah. Yeah,
Yeah. That's why I was saying. Sorry, go ahead.
the interesting thing is that that when you imagine right at the Big Bang, as things, the first rapid expansion, that there were these areas that were denser than other areas. So there was
Yep.
it wasn't homogeneous. It wasn't pudding. It was like a heterogeneous mass that basically had some masses that there were some massive or dense areas, and those areas could have
Hello,
quickly
sweet
collapsed
internet.
into black holes. So
Right, and it's probably good to note that when we say massive here, we're talking like a hundredth of a thousandth of a percent more massive or less massive.
right,
So
yeah,
so pretty homogeneous, but it's
yeah.
also not entirely homogeneous.
Yeah, and it's interesting to think about what caused that, basically.
Right.
But then I think that just work like random statistics. That's what would happen.
Yeah, we're pretty sure it has something to do with like quantum fluctuations early on, but there
Yeah,
are a bunch of different models kind of to how they ballooned up in size, so to speak, to what we see
yeah.
now.
We got a
Because
kitty here.
they're the spots where the galaxies formed, quite literally.
I have a few seconds of video for to show what the idea of what like the early universe, how it might have looked like in a 2D sense with a
Okay.
record
Say hi, Loki. Loki says hi.
spinning really fast. It would turn into like you see how these big chunks and stuff.
Hold on a second. I don't know how to make you big on this. Uh, I'm not entirely sure. I don't
You
know how
can
to
see it
use
just fine in my window.
Have you ever used Restream, Top Dog?
When it first came out, maybe,
Yeah, well, yeah. I wasn't impressed
and
the
then
first time I signed on to it. I don't know, a year or two ago, and I rejected it. But now I really like their user interface.
Hmm.
They have a lot of stuff that's pretty cool. This chat overlay thing that shows you the new chat on the right automatically. That's kind of neat.
Yeah, that's
So
kind
I
of cool.
could basically display the chat and let people, so that I don't have to keep highlighting everything.
Oh, that's cool.
Yeah, that's not bad.
it
And
does have a
I
lack of
think,
options when we're looking at it through this uh, on my end. Like has like the window is very small when I'm looking at the chat on YouTube. I have like,
oh yeah,
and then it has already on the restream has its
you
own thing,
can
and you can't remove it.
pop the
So then it
chat
makes
out.
it harder.
I think you can pop the chat out.
You
Let
can't
me see.
respond on the chat on the restream, so that's why I have to use the other window from pop out of YouTube. So then it makes
Oh
this window
no,
smaller.
I couldn't respond on my first stream, but I can now. From Restream, I could type chat.
And it puts the like video on the left instead of underneath like StreamYard does. So it makes even
Video.
the video smaller yet. But the private chat's good where it's underneath. That's the only good part.
Yeah, well, I got a 55-inch monitor, so I'm not too limited in how big I can stretch this thing. So,
If I make it
uh,
full screen, I'll lose the pop out chat. Unless I want to cover half the
yeah,
video.
yep.
So
Three
you have all
six,
those problems.
3 out of
No
6 people.
editing, the
I can
widgets.
have 6 people for free. How about that? I'm going to sign up with these guys, but I'm waiting until because I'm going to cancel StreamYard because I'm tired of the echo. So far, we haven't had a problem on this, and this works with Firefox, whereas StreamYard, I couldn't get even Firefox or Safari or anything to work with audio on my Mac. Right now, I just can't use Google Chrome. So anyway, Scott was going to join us, but what else we got, Andrew?
Right now I'm trying to find my original source for what I just said.
Oh
Here,
yeah, put that in private chat
I'm gonna give you
or some chat.
yeah. So this isn't the yeah, private chat. So this isn't the original place I found it, but here's one where I just found it. Let me find the other one.
Oh shit! Hang on a second. I got to mute for a minute.
I have the site that you posted on my screen.
Okay, I will put that in. Copy link. What?
Where's the other one?
What's happening? Okay, so that's the paper.
So that's from space.com. I'm trying to find the one that had the paper linked with it.
Put that in there. Let's see. Confirm James Webb Telescope. Let's see here. Put that up. Get rid of me. It's not about me. Not all. Always me. Fucking hate that.
You are the host.
I hate me.
Okay.
Later. I will subscribe later. Just sure I will.
Subscribe. What?
I don't know. Wants me to subscribe to this thing. What
Darn it!
is
I can't find where I originally saw that. I'm sorry.
I don't know
I'll
what
have to
is
look later.
happening right now. I don't know why. Oh, it has that. Okay, now you can see
Is
it.
it harder than StreamYard where they change the window sizes?
No. No, it's just that they've got like all these different options, and one of them is they have like all these weird options for layout, like that, and that shows me half of my window, and this one shows the whole thing, and this one gives you a nice little. I can put you guys at the bottom and show the paper at the top, which is actually kind of nice.
Hey, Speed, do you want to pin the link? I see Jay's in chat. Hey, Jay.
I think I did.
Oh,
I
you
didn't
did?
pin it. I didn't pin it.
Yeah, in case someone else pops up.
Yep. Yeah, because we don't know what the hell we're doing today.
We never know what we're doing.
No, we never know what we're doing. We're professionals. Professionals don't know what they're doing.
I did like one part they said in this article, actually, they said or according to the astronomer doing the research, that, like I said, the properties of the stars that they measured from these early galaxies were matching what we thought they should based on our current galaxy formation model. So I thought that was interesting.
Yeah.
I guess confirmation is always good, but it's always disappointing sometimes because it means what we thought we knew was actually right. I guess. Yeah.
Look at this little.
You know
look
what I
at
mean.
this. Look at this little ad here. Now legal in Minnesota. A pain reliever so powerful. Do you know what that pain reliever
What?
is?
It
Edibles.
looks like a berry.
Actually, it's loaded with THC. I guess it's pretty fucking powerful too. But they made that legal in Minnesota. So revealing
So,
the first
Andrew,
galaxies.
Basically, science isn't always perfect. It gets closer to accurate to reality for our understanding.
Asymptotic. It is asymptotic.
Yeah. I was just saying, like, sometimes it's disappointing that what we thought we knew isn't actually wrong, because that means there's not as much to discover as we thought it was, or at least initially.
Yeah.
Because, like, if you're wrong about something, that means, oh, hey, we can revise our models. There's so much more to learn about now. And the good news is we have, or actually with James Webb, with all the mysteries we have left to solve. So that's always exciting.
Yep.
I've heard that scientists love not knowing because
Yeah,
there's always something to
in a
learn.
real sense, yeah.
Oh, Sherwin
If you know
Williams
everything,
paintings.
then there'd be nothing to learn.
Yeah, exactly.
Somebody wanted us to know that Sherwin Williams paints are 30% off until December 22nd. I love these kind of websites. I stay away from this shit normally, whenever I can.
Yeah.
Anyway,
Yeah, like I said, I tried to find the one with
the paper.
Hmm? Yeah, I tried to find the one with the original paper, but I couldn't find it.
Yeah.
Like, I saw it yesterday, and now I can't find it.
Well, if we get it, there's a little video here. I think. Click here for mail. Come on. Oh, I got a beach ball going on here. What's
And
happening?
you put this in the description, right?
I did. Yeah,
Okay.
it is saved. I got it. Pretty cool.
We are saved.
We're saved. Oh, let's see. Stephanie doesn't read and she doesn't do maths too. Also, you should have said also. Apparently, you can't write, Jay.
Got him.
Gotcha. Yeah, fucking zing. Damn it. Nailed his ass. Uh, anyway,
Math is all out of context. What does that even mean?
I don't know what
Stephanie,
the
you
fuck.
confuse me sometimes.
Yeah. Yep. Anyway, so Kafai, our psychedelic resident sea god, do lots of mushrooms and sea god friend, he keeps talking about Johns Hopkins, and there are two researchers. Uh, one is Richards, and the other one is Roland Griffiths. And Roland just got diagnosed with terminal cancer. And some of the work he was doing at Johns Hopkins was giving psilocybin to people that were terminally ill. And lo and behold, now he's terminally ill. And I think he finally started entering into some psychedelic experimentation himself, basically, and dealing with his own life and death and everything. And I should link that. I have it's a really interesting podcast with somebody named, maybe somebody's heard it, Tim Ferriss Show. Does anybody know of the Tim Ferriss Show?
Nope.
Anyway, he's pretty cool. He does a really good job of interviewing. I'll put that in the description. But I was kind of sad. I picked on Roland and Richards because sometimes they seem a little woo when they talk about psychedelics. But in this interview, he lets on. He says, "I don't believe in reincarnation or an afterlife or anything like that." But he talks about how it's something basically. There's nothing it's like to be dead, but that is like something, right? I mean, to be completely like annihilated, it is like something, right? To not be there anymore. And what's really interesting about the end of your life, and this is really cool. So at the moment you are no longer conscious, okay, you're no longer anything at all. You're not even unconscious. You're not sleeping. You're not anything. At that moment, if you were to look ahead 300 years, it would be exactly the same as the moment that you're no longer conscious, right? So, but look ahead 300 trillion years, and it is exactly the same as the moment that you died. So when you die, in some respect, the sense of you at that moment is the same for all of eternity. So that is a nice way to look at eternity. I mean, it's annihilation. You aren't going to be sitting there saying, "Oh, I'm dead." You're not going to be doing that. But
right.
But there's something to it. It's really kind of cool to think about it. Hi, Nancy.
About the
Hey, Nestle.
13.8 billion years before you were born, before you had a consciousness.
It's the same, right? It's the
You don't
same as
remember any of
before
that
you were
either?
born. And that's what it's like to die. It's like just like it was before you were born. And you weren't freaked out about the time before you were born, right?
Right.
You're not going to be freaked out when you
That's
die.
true. Yeah.
Yeah.
I never thought of it like that.
Yeah. So when
Can
you
anyone
die,
hear
you
me?
just don't. Yeah, we can hear you.
We're just afraid of the. Oh yeah, we're just afraid of the pain and suffering that will maybe think of when we're in the last moments of our consciousness and stuff.
And that turns out to be pretty well managed in modern medicine, so that isn't really a problem either. It's just that I'm not gonna have any more experiences, and but the thing
brain
is
has its
that
own safeguards too, and that's why we have afterlife people are talking about their experiences of their dying brain, whatever.
Yeah,
but
you
that
probably
comes back sometimes, so they can tell others about how their dying brain was given this euphoria.
Yeah, they basically sounds like a neat way to get high. I don't know, die, your brain will start shutting down in sections and there'll be all kinds of things happening, like the light tunnel. Okay, it's a natural characteristic of your occipital lobe and your visual system that when you start to go unconscious, your peripheral vision and your vision becomes narrower and narrower so that it looks like you're in a tunnel or a well. And then it kind of like narrows down to a point of bright light and then it goes blip and it disappears. So you naturally see a white light at the end of a tunnel based on the anatomy of your visual cortex, which is kind of cool. So you get to see the tunnel of light. What do you want, Siri? I don't know what you found. I don't care what Siri found.
What have you found for us today, Neslig?
Oh, I unfortunately got late in because I really scrolled backwards in the live stream and I saw that Speed shared a very interesting paper on flowering time. Really.
Oh.
And I missed that. Sorry.
Oh, you mean the Augie paper, the pleiotropy paper? Yeah,
yes.
Yeah. I linked. I put the title in the chat. I've got the title, and if you type that in, you'll be able to look it up.
Is it about the APC system of flowering?
Yep.
Flowering development.
Yeah, I think let's see, where are we here? Trying to find that.
The Matchbox genes. Yay.
Yeah, Madsbox. Yeah.
Mm-hmm.
Yeah, they talk about Madsbox genes.
I thought you said Maddox for a second.
No, it's Mads. Instead of Hawks, it's Mads.
MADS, Madsbox.
Okay.
Yeah. Not Matchbox either for like cars.
Yeah, let's see if I can pop it up here.
It's a plant equivalent or analogous to the Hawks genes of animals.
Yeah.
Oh, I didn't know that.
They are not homologous. They are very different genes, but they do regulate, or at least we think they regulate gene expressions.
Yep.
Okay.
They are like I think they are transcript factors, if I'm not mistaken, or maybe they are indirectly involved in regulating gene expression. But yeah.
They get into the basically pleiotropic constraints on evolution and ways around it. Basically, the creationists would say, "Oh no, you can't have that," but no, there are ways around it, and it's
very interesting that many genes are just used over and over and over again, very different things, and I think that's one of the way why we have a pleiotropy effect of many different genes. But then the interesting thing you can have is when you duplicate genes, that one set can be specialized for one function, another can be specialized for another function. That reduces the pleiotropy also.
Yep.
Right. Yeah.
Yeah,
that's what you were talking about earlier, wasn't it, Speed?
Yeah. And the other part of that is the reason that genes will end up being used over and over again is as those cells differentiate into their own subtypes of cells, and you keep going down that hierarchy, right? Depending on what the shape of the chromosomes are, and what the regulatory effects are, a lot of those things are still going to be operating, and they're going to fit into different pathways all the way down. So and some of the pathways that end up being really important, like the Wnt beta-carotene pathway, is very, very important and ends up being conserved across all animals. And it's just, it all fits so naturally into evolution that the only way to make sense out of it is with evolution, basically.
And specifically about the Matchbox genes, like I believe, like the early relatives of plants, like also the green algae, also have some Matchbox, but they function as individual proteins. And later on in the evolution of land plants and specifically flowering plants, you have repeated gene duplication events. And now the Matchbox, they form a complex, a tetramer, with 4 individual proteins, which is really
So
interesting.
kind
It's
of
almost like,
similar to the Hox gene duplication.
well, it's I don't know if Hox if the proteins of Hox genes also form complexes. I'm not familiar, but if you look up an image on probably like a Google image probably will show you that the proteins themselves of Matchbox they form this tetramers. This a complex of 4 proteins.
Oh, okay.
Yes, and the original genes in algae, for example, they do not form such complexes. Let's see. There is no image. Can you perhaps Matchbox complex or tetramer perhaps Google that speed? Maybe that will give the image. Yes, the one on the top left, right there. Yeah, that's a good one.
Yeah, right there. Okay.
Yes, you can see. This is the ABC. The classical system of the flowering development is the ABC system. Although sometimes you have the ABCD system. So as you can see here, basically you have different types of Matchbox genes. The A set involves in the development of the first, second row, which are the sepals and petals. And the B set involves in the second and third row, the petals and stamen. You have if you, for example, if you express the A set and the B set, that will give petals. But if you only have the A set, it will give sepals. And if you have the B plus C set, that will give stamen. Like basically, it's a combinatorics of the different set of genes.
Yeah.
And then they are believed to produce these complex of 4 proteins. As you can see here, you can see in the image that the 2 blue circles and 2 purple circles. Like that's a set of 4 proteins that combine in this way.
Interesting. Yeah.
And they are all homologous to each other. They are the result of repeated duplications. And the original Matchbox was just 1 protein, which doesn't do this. It doesn't form these 4 protein complexes.
There's
and this is my wheelhouse because this is my at least part of my master's education about plants.
Oh, nice.
So I can this is one of my rare opportunities I can nerd out over plant biology because plants get ignored, unfortunately.
They get ignored, and evolution is so easy to see in plants. And Scott. Hi, Scott. So in plants and even fungi or fungi. I never know how to say that. Evolution is a lot easier. Fungi, yeah. Evolution is a lot easier to see than with fucking mammals.
Yeah.
And it's much more common. And if we were able to go out and watch the woods for 10 years, basically with insects, plants, and fungi, we would see all sorts of speciation and evolution and basically beneficial mutations and all sorts of things. If we had the ability to keep track of the woods is a very complicated place. As I found out when I was 11 or 12 years old, I took a 1-foot square section of the woods in the middle of the Minnesota winter, and I brought it into my room and set it on one of my mom's baking pans. And my mom was screaming because insects were hopping off that thing and jumping downstairs and spiders and all kinds of shit. It was amazing when it thawed out. The woods is very complicated. Just a 1 square foot of dirt ended up being this biological mess that my mom did not appreciate. So she put me in a home. No, she didn't. An orphanage. No, she didn't. But when she came home one time and I had a calf head cooking on the stove, because she came home early and she surprised me. I was boiling a calf head so that I could get at its brain and dissect it. And my mom came home and found it boiling in her soup pot in the kitchen. And that was the point where she put me in an orphanage. So it took a lot to piss my mom off. Anyway.
Hey,
So, yeah,
Scott.
this is some cool shit. How's a trumpet lesson?
I have another. Oh, if you are interested, I have another good image to summarize the evolution of these genes. If you're interested.
Okay. Yeah.
Can I in the link? This is a manuscript. I think it's published, but the manuscript has also the image. But if you see, can you see the link in the private chat?
Yep, I do.
All right. And if you go down to figure 2, it's a very good figure.
What in the fuck am I looking at here?
It's a manuscript.
Let's see. 14,400.
It's a PDF, yeah. And you have to go down all the way down to the figures, and it's figure 2. It's a very good figure.
Let me find it. Are the figures on the bottom?
It's past. Like manuscripts often put the figures past beyond the citations. Yeah.
Here's figure 2.
Okay, you zoom in a bit, perhaps, or not? Make it bigger.
Let's see. Could I zoom in? Well, I could try and see what happens. Yeah, that works.
All right.
Okay, now we'll unclip it. How's that?
Like to me, it's a very bit blurry in the
Really?
studio, but I don't know how it looks like in the live stream itself.
Yeah, I don't know.
Also.
Let me go check. Yeah, it seems a little blurry. Is that because? Well, that's because I'm not paying for this thing. That's why it's blurry. Yeah.
I can put a link in the public chat for anybody to see it clearly. But what this shows is basically you see the homologs of these genes also in the green algae relatives. And in the green algae relatives, if you look at the B section on the right, which gives the matchbox genes, in the original, it's there's only there's only one domain. So it forms a dimer in the green algae. In the relatives closer to flowering plants, for example, in the gymnosperms, it begins to form dimers and tetramers, which basically in them these matchbox genes induce the development of cones, either female cones or male cones, it makes sense because it's the same. It's the sexual organs, just like a flowering plant. But then when you go into the flowering plants themselves, it gets even more complicated with more additional duplications. It's a very good summary of the evolution of these genes.
Yeah, and as far as proteins forming dimers and tetramers and all of that turns out to be ubiquitous in proteins.
Right.
Proteins stick to each other.
It's also the case with our hemoglobin. Hemoglobin is also a very interesting evolutionary. Like our hemoglobin, like we have as after birth at least, we have the 2 subunits A and B, and each hemoglobin has 2 of each. So we have 2 A subunits and 2 B subunits comprising the hemoglobin complex. And those subunits are also related through the gene duplication as well. So yeah, the original was a homodimer where you have only one subunit forming a 2 subunit protein, but then after duplication you have a heterodimer, a protein of 2 subunits, of 2 different subunits, and then later a 4 subunit complex with further modifications. And they are also related to myoglobin. And interesting still, like as placental mammals, we like as embryos, we express different hemoglobin genes that are more, because as as we develop, we as a fetus, we need to be able to draw up oxygen from our mother, basically, and that requires a stronger hemoglobin, basically. So we need to express a different hemoglobin that has a higher affinity to oxygen, and then we express different hemoglobin genes, which are also again related through gene duplication.
Yep. And I'd like to take this moment to do a little Cell Cordova bashing. He had a recent video about how impossible it is for there to be dimers and tetramers of proteins, that they had to be designed just properly so that they would stick together. And it turns out I have one paper that talks about, I think 80% of all proteins kind of naturally stick together to some degree, to varying degrees. But dimers and tetramers are ubiquitous in proteins. It's very, very common for that to happen. Yeah. It's because of the 20 amino acids, there's only 4 real characteristics. You know, your acidic, basic, your polar, and non-polar. And they tend to complement each other and stick together as dimers and tetramers.
So when they say design, that just irritates the frack out of me, because all they are doing is inserting yet another blank space to stand. It's like getting one of those redacted documents from the government, and more than 90% of it is just these black marks across the words. It's like they're saying, don't ask questions, because you're gonna, they say, well, this was designed. Okay, who designed it? When was it designed? At what point does it stop being natural processes, and was this particular piece designed in? Because they believe the whole thing is designed, right? So therefore, the word design means absolutely nothing. So I don't know. You just can't be scientific.
It's a non-answer. It's a non-answer.
Yeah.
There's no substance. Like if you would tell them, oh, it evolved, they wouldn't accept it as an answer. But we should expect them to accept, oh, it's designed as a sufficient answer.
It's just filler word.
It's a double standard, basically. If evolution cannot answer 100 questions out of that they can ask, or even if you can answer like 50 out of 100, then it's still not sufficient. You need to answer all 100 questions. But then for design, you only say, oh, it's designed, and no more questions asked. That's basically it. Yeah.
Yep. And the interesting thing, when they start talking, like when you start arguing with them, right? They'll basically, there was 4.5 billion years ago, all the way to the present, okay, where things like come from the zygote by purely naturalistic chemical means and turn into full organisms. And then you look back a little bit and you say, oh, well, did evolution happen after Noah's Ark, where the dogs become, German shepherds and whatever, and they will agree with that. But then when you try to argue with them about any one point along that line, they will shift all the way back to 4.5 billion years ago. And then when you argue about that, they'll move ahead 4 billion years into the future, and they don't think properly, right? They're thinking about like 6,000 years in their head. And it's just really bizarre to try to argue with them, because they keep moving the goalposts back and forth, and you can't get them to land. It's like, okay, if I grant that God created eukaryotes, or created the first multicellular organisms, and everything he did, are they denying that everything evolved from then? But forward, that is the part I want to talk about, and that's where we're talking about evolution. And that's the thing they do not understand. Every time you argue with them about evolution, they will try to argue with you about abiogenesis. And they won't fucking stay still long enough for you to get a point across. I'm looking for Stephen Colbert did a thing this morning on his opening, or last night, his opening dialogue. And he always does You know, Stephen Colbert asked people to suggest names for Trump because Stephen Colbert has not uttered the word Trump for 2 years now, I don't think, or 3 years, something like that. He always says the president, and he always
yeah.
basically
I
cuts it
notice
out.
that every time.
Yeah, but okay, what was the name he had?
I
So
like
anyway-
I like Aaron's name. Marmalade Mussolini.
Marmalade. Oh, I hope he sent it to Stephen Colbert because Stephen Colbert uses a new one every night or for every opening monologue. Okay. So anyway, he asked users to send it in to Twitter, I think it was, and they saved them all. And every night he uses a different one. And last night it was the white scrotus. So it's right here. Where the hell is it?
Oh, also, like what you said about proteins, speed, that also reminds me of another paper that I linked in the private chat. It's it says that proteins, like I said, they form complexes, and they also form these geometric symmetrical structures. And it turns out that the reason why proteins have the tendency to form these structures is because it takes less information to do so. So it's basically they have an inherent tendency to form these things instead of it's basically the opposite of the design argument. The design argument would say like, oh, it requires a designer. It takes so much information to make these things, so therefore it requires a designer. But it turns out no, that they have the innate tendency to do so automatically. It turns out. Yeah.
And if you want to go all the way back and look at the origin of all those proteins in the protein orchard, they were a fuck of a lot simpler, basically 4 billion years ago, than what we're looking at now. And that's why it's not a problem for evolution.
I can read from the abstract. It says, why does evolution favor symmetric structures when they only represent a minute subset of all possible forms? Just as monkeys randomly typing into a computer language will preferentially produce outputs that can be generated by shorter algorithms, so the coding theorem from algorithmic information theory predicts that random mutations, when decoded by the process of development, preferentially produce phenotypes with shorter algorithmic descriptions. Since symmetric structures need less information to encode, they are much more likely to appear as potential variation. Combined with an arrival of the frequent mechanism, this algorithmic bias predicts a much higher prevalence of low complexity, high symmetry phenotypes than from natural selection alone, and also explains patterns observed in protein complexes, RNA secondary structures, etc. So yeah, it's a really interesting story to read up on. I can put the link in the live chat as well. There you go. Hello? I don't hear anybody anymore. Oh,
I hear you, Netzlik. I hear you, man. I was going for that microphone button as fast as I could.
Let me put that in the description. Where are we here? Okay, guys. Oh my God, that's a big link. What the fuck? Why is that link so big?
Oh, I don't know.
It's a gigantic link. Why is it? Oh, redirect token. Redirect token? What the hell is going on here? Let's see. I don't know what I just did. I'll figure it out. Oh, it's a small link, actually. Control C. Control V. Okay. Let me pop that up. And turn the camera on. There it be. Symmetry and simplicity spontaneously emerge from the algorithmic nature of evolution. Cool. Is anybody still here?
Yes. I'm still here.
Okay.
You guys got abandonment issues all of a sudden.
I have abandonment issues, man.
I was busy doing other things. Sorry.
Anyway, okay, that's in the description. So yeah, this looks like an interesting read. Let me go ahead. This article hasn't been updated. Oh no. So Scott, how's it going with the trumpet?
Well, it was a good session. It was just like a 30-minute over the phone thing. He was just the instructor was just giving me some new assignments to do and changing my practice schedule a little bit. It's sort of like going to the gym, and at first you start out real slow, and then they start loading you down with stuff to do, exercises. So that's kind of what happened today.
Yep. He keeps messing with you.
Well, that's what an instructor is supposed to do.
Yeah, well, it kind of sounds like my psychologist. He keeps giving me assignments, shit like that.
Yeah, that's what they want you to do to improve.
Yeah.
You have to do something new. You're not going to improve by doing the same thing the same way.
But you didn't like my idea of giving all of your possessions away and taking your dogs and going across America with your trumpet.
No, I loved it. I loved it.
Really?
Yeah.
Are you going to do it?
Probably not, because I'm lazy and scared.
Scared.
Hey guys, I need to. I'm going to do to the gym workout for today, so I will be talking.
There you go.
Next time.
Okay.
So,
Yeah, Nestle.
thanks for having me.
But, Nestle, will you? I sent you an email today because I forgot something that we were supposed to talk about, and I forgot what the fuck it was.
Yeah, right.
Yeah, okay. Yeah. So please.
Sure. All right.
Please.
All right.
Okay.
I'll see you later.
Respond. Okay. Bye. Anyway, so what else is going on? We're gonna have a big snowstorm. So, did you have you had a tornado in Fort Worth, right?
Yeah, there was up north of me here. They did have some touchdown and damaged property. I don't think there were any lives taken, but it was just one of those mornings where you wake up to one of those big, big thick front lines of storm just blowing through.
Yep.
Dogs, they curled up right on me and just wanted me to comfort them.
Well, it's been straining here. Straining.
Snowing and raining?
Yeah, at the same time. And apparently we're going to get, the temperature is going to drop low enough tomorrow that we're going to get accumulation of snow. But mostly we have accumulation of a lot of slippery sludge everywhere. It's pretty intense. Pretty cool.
So you talk about the weather, and the Weather Channel saying that there was a hospital that was damaged.
Yeah, in Fort Worth.
In Louisiana. So
I wonder how I make somebody full screen. I don't know how to do that. With
how'd you do it for yourself?
I'm the first one I'm in the top, so anytime I do this, it takes me.
Drag?
I could drag you, yeah. Yeah, that would work. But anyway. I think I have to move you. Okay, yeah, I can move you. Yep, so that works. So that's how you got to do it, I guess. Anyway, we'll figure it out. So what else?
There's nothing cooler than getting a science-related tweet from like somebody like Gutsick Gibbon or somebody like that.
Did you get one of those?
Yeah, it was just she had retweeted some article by somebody saying that hominins learned to walk upright in the trees.
Oh, nice.
So
there's also that bit about fire being used by non-hominin species that came up recently. If fire was used a lot earlier by other primates other than
well, I knew there was one that showed that Homo naledi had used fire. But no, I didn't hear one about videos of non-hominin primates.
Hey, Rage to Reason is here.
Yes, Rage to Reason.
What are you saying, Top Dog?
Oh, didn't Gutsick Gibbon already posted videos about the things you were talking about?
The Homo naledi, yeah. that
Yeah.
one, but the one about walking upright in the trees, I don't think I've lately seen a video about that. But she was retweeting somebody else's work.
Yep. Psychologists, that's what they do. They give you assignments. So what are my assignments this week? Further. I got to go further. That's my assignment.
Get
Go
rest.
further, go deeper,
I have to go further.
Keep
further.
living.
go further into
Did I link?
the subconscious.
I wonder if I linked that Roland Griffin's thing. Cosmology links. I did not. Maybe I did. Who knows? No. Oh yeah, the podcast. Yeah, psychedelics can help, and the art of living a life of gratitude. That's what he's talking about. Roland Griffiths, who I feel bad about giving him all the shit I gave him, because now the guy's dying. And it turns out he's a cooler dude than I thought. Now that he's dying, he's coming out and basically he doesn't give a shit what he says, because he's no longer worried about his grants, right?
Indeed. Who are you talking about?
Roland Griffiths, the lead researcher at Johns Hopkins on psychedelics, where they're doing all that work on PTSD and addiction and end of life help for people that are dying, basically helping them get accustomed to the idea of dying.
Well, yeah, if he doesn't really care about grant money and he just wants to speak his mind, that's a pretty good thing.
Yeah, so he started actually sampling what he's testing, basically, and he couldn't do that before.
Oh,
and he sounds like a really cool dude. Like, he was talking about going into surgery and getting this procedure to install a port in his chest for chemotherapy, and people were like, "Oh, that's horrible," and he said, "No, actually, it was kind of fun." It was kind of fun. It's like,
Be like Iron Man.
yeah, but he has just an unbelievably positive attitude and laughs about his diagnosis and stuff like that. And it's a pretty amazing guy, actually. I link the podcast in my description, so I think people
I'm
should
gonna
listen.
check that out. That sounds interesting.
It's about it's a long one. I forget how long it is. I think it's, let me see here. It's over 2 hours. No, it's 3 hours. 3 hours long, but it's just a good talk. The whole thing talks about how he got into the research and his past and all sorts of stuff. So highly interesting. What else? What else are we doing, guys? Can we get out of here now, so I can go make a steak?
Sure.
I got two big highly marbled ribeye steaks.
Been wondering. It's been over a year now that my website's been up for Science and Rationality.
So how you?
No one has been really doing anything all this time.
It takes
for people to help.
It takes a long time to basically get a website going.
It took only a few minutes for me.
So
No.
Because I already had your website, I guess I added it to this.
So
To
Speed,
give
is
you-
it that how.archives or is it that padtoppin.se podcast? Which one's the guy talking about psychedelics?
It's the big long link. Let's see. Pod to open or whatever. SE.
Okay.
That one, yeah. Let's see. God,
It's on the Tim Ferriss show? Okay.
Yeah, and I don't understand what all the other shit. Roland Griffin's PhD, life's ultimate glide path and unexpected stage 4 diagnosis, facing death, how meditation and psychedelics can help in the art of living. So it's pretty good. Yeah, more people should check out your website. But what I'm saying is, it takes a long time for a website to gain traction with people.
Oh yeah.
So it doesn't happen overnight. As my YouTube channel is evidence that it doesn't happen overnight.
Yeah.
It takes people sharing, and then maybe advertising in ways that
Yeah, I
We
usually
don't want
put
to
your
spend
link.
money on though.
I usually put your link in the description, but I've been lazy in many respects lately. So anyway, guys, I think we should shoot it ahead. And okay. So there's a big that button end stream. I couldn't see it the other day because it's like a dark blue and or gray, and I don't know. It just didn't register. I couldn't get up.
Oh.
But I think I know where it is now. So now we can end the stream. But when I end the stream, we don't have any closing, nothing. We're just going to shoot it in the head, and it's going to end abruptly. It's going to be like our lives. They're going to end abruptly. Oh, that's a dark thought.
Well, stay
What do you
curious.
say? Okay, there you go, Top Dog.
Bye.
And goodbye, cruel world.